organizing the user organizing committee. I am the representative for structural biology, and it's a pleasure to introduce Tilman Grunewald, who was the recipient of the Young Investigator Award last year at the SRF, and who is going to present you the seminar of today. Tilman, the floor is yours.
Thank you. Well, hello. It's nice. I mean, I don't see you personally, but I see a lot of familiar names. So, hi to everyone. Today's seminar is part of a whole webinar series that the user organization is organizing. So, stay tuned for that. Today's guest is ID11, and John Wright is presenting his beamline. John actually joined the ESREF in 2001, first as a user and then as a postdoc, and he basically stayed on at ID11, where he's the principal scientist now. ID11 is actually one of the first beamlines of the ESREF. That's something I learned back in the day. It was built initially by Ake Quick, then led by Gavin Vaughan, and then I think in 2014, John took over the beamline. In very general terms, it's a material science beamline. It's about material science, high energies. One of the hallmarks of ID11 is the combination of high energy and nanofocusing capabilities to do any kind of crystallography, and crystallography combined with tomographic imaging. But I'm not going to spoil more of John's talk, so let's give the floor to John and let's hear it. Thank you.
So thank you, Tilman, for the kind introduction. Thank you for STREAMLINE for organizing and helping us with this webinar series. I'm hoping that everybody can see my slides already. And so you should be looking at a photo of Pierre-Olivier and Haijing, and they're crowded around our 3D XRD diffractometer there, and they're carefully adjusting this little micro stress rig, this Nanox device, which is about to be inserted into a furnace. So right now, as I speak, over on the beamline, Haijing is running his experiments, and he's got samples in the furnace at high temperature to see how they behave. So I should make clear that this talk today is me being the spokesperson for a whole team of people that do the actual work here. So the first thing I wanted to try to do is to give you a quick virtual tour of the beamline. So ID11, as Tilman said, was one of the first beamlines at ESRF, and so this optics hutch here, OH1, was one of the first hutches on the experimental floor, and now it starts to show its age a little. The next piece of beamline that was built was this EH1 experiment hutch. What this is used for today is large kind of heavy duty sample environments. So in this hutch, there's a nice big space where you can bring in sample environments. And the great advantage here is you can kind of come in and do experiments that might not fit somewhere else. The limitation is we don't have the highest spatial resolution. It's only about 10 microns, but you can have this 150 kilos. So then as we move along the beamline, there was a EH2 hutch here, and this is where the first 3D XRD microscope was built in around 2000. That was collaboration from the DTU group at RISU. That instrument was moved out to make a long beamline. So just before the phase 1 ESRF upgrade program of making long beamlines, ID11 and ID13 were both made into long beamlines, and this 3D XRD station was moved out to be further away from the source. So this takes samples up to about 5 kilograms, and now we can reach around 1 micron of spatial resolution. Our latest station, this nanofocus station, was installed in 2016. And with this station, we can get down to 0.1 microns, that's around about 100 nanometers. But now the sample size is limited to 500 grams. So we have this suite of diffractometers that are trying to map onto different sample dimensions. So I think the biggest news for many people at ESRF recently is the upgrade of the source. I like to think that this whole EBS project was done essentially for ID11 and to improve the source for us. So this is the graph that was in the book describing the project, and this green curve is the spectrum from one of ID11's undulators. And when the source was upgraded, it went from being this green curve to being this blue curve. So all of these photons that we weren't using that were in between the undulator harmonics got kind of swept into the undulator peaks. And we now see something like 40 times more photons coming down the beamline. So to be ready for those photons as part of the phase 2 upgrade, the beamline was equipped with a new suite of detectors in EH3. So we have a very fast Eiger cadmium telluride detector, that's photon counting. There's a pair of scientific CMOS detectors for all of the diffraction imaging and imaging experiments. And there's a new microscope to give better quality pictures. So what was the first thing we did when we got the beam from the new synchrotron source was just to check how much flux there was and if there would now be sufficient flux for everybody to be able to do everything that they would like to do. So Eleanor was our postdoc, and she arrived during one of the brief windows when you could travel during COVID, and arranged to put different sample geometries on the beamline. This was in monochromatic beam, so we're putting the beam through the monochromator. And as you change the sample geometry and you change the incident flux on the sample, Ellie pulled out the thermal expansion of the sample, which we assumed was due to heating up. So during these experiments, the flux is gradually increasing, and we measured how much the lattice parameter is changing in the sample, and she optimized the geometries to get the minimum possible heating and kind of to increase the heating as much as possible. And then the real proof of this, and this was quite stunning the day that it happened, was if you got the sample geometry just right and you stopped as many photons as possible and you thermally insulate the sample, then when you turn the beam on, you go from a fully deformed diffraction pattern with nice smooth diffraction rings that the copper heats up sufficiently that it recrystallizes in the beam. So this is what happens when a plumber is welding the pipes in your house and you eventually get leaks because the copper's recrystallized. You can do the same thing with the X-ray beam. So Eleanor developed a detailed model of these different processes and how this was happening. And so in practice, we normally have much less absorption in the samples. We spread the beam over a larger area, or we have better thermal conductivity for cooling. And it's not normally a problem. But if you do need a lot of flux, we usually have sufficient flux for most things that people want to do. So what kinds of diffraction experiments do we do? It's anything you can imagine with X-ray diffraction. Probably someone has done it or tried to do it ID11 in recent years. So there's a whole suite of single crystal diffraction methods. There's a suite of doing powder diffraction methods. And then there's this everything in between, which is spotty data, multi-crystal diffraction, 3D XRD, diffraction tomography, and so on. So we have ways to change the beam size. So you can have a very small X-ray beam. You can have a slice of X-ray beam to take slices through the sample, or you can have a large X-ray beam to illuminate a volume, and you can also use that for tomographic imaging. You can then collect the diffraction detector with a near-field detector, so the detector very close behind the sample, and you see directly the shapes of the grains, or with a far-field detector that's very far back, and you see directly the scattering angles. So what I'll focus on first with the kind of science people do is on these single crystal methods where there's just one single crystal in the beam, or we aim to get one single crystal. So this is a selection of structures that have come out recently, and I think most of them were determined since the ESRF EBS was upgraded. So the good quality crystallography to be able to solve and refine difficult structures, I think is the foundation of everything that we do at ID11. I think each of these publications would deserve a much longer talk in itself and to go through the reasons why and the things that were learned. But the main message that I want to pass is that you can get good quality structural data from very tiny crystals. There's another point that I think is important is that I think all of these groups are using electron diffraction and electron microscopy to look at their crystals as well. And the role of the X-ray methods is to get the really accurate structural details about the chemical bonding here. And the small crystals is an important part of what we do, that you can measure very small crystals. And I think electron diffraction or electron microscopy has been key to getting those crystals onto the beamline and selecting the samples. So a method that's been developed and is a very exciting development for us at ID11 is measuring samples inside of diamond anvil cells. So this is an approach that was developed by Leonid and Natalia Dubrovinsky. They started coming to ID11 when we first had this nanofocus station. And I mean, they really worked hard and spent a lot of time to get this method to work. But now they have a really effective workflow where they put up a diamond anvil cell. I should make clear that on ID11, they just put up the diamond anvil cell and it already has the sample in it, and all of the difficult things of changing pressure and making the sample have already been done. They make an image of the sample by scanning the X-ray beam across the sample and do a powder diffraction snapshot at each point as they scan across with the beam. Then when they look at this data, they're sitting live at the beamline and they'll decide, okay, I want to measure the single crystal data for this specific crystal here, or for that specific crystal there. And they can go and choose a crystal from their diffraction map. And with the instrument that we have, you can align that crystal on the center of rotation to measure the 3D crystal structure. And I think the reason they like to come to ID11 is because the instrument allows you to put this crystal on the center of rotation. So that workflow of doing a 2D diffraction map, selecting a crystal and do the rotation scan is very, very productive. And there's Dominique Laniel and the Bikoffs, Maxime, and they have their own groups now. And it seems to be a growth area because you can do fascinating chemistry at these extreme conditions. So again, I've put up a selection of papers where people have solved, refined very difficult crystal structures in very difficult conditions using the instrument ID11. I won't try to explain the details of what they did. So now if I come on to the 3D XRD methods and the grain mapping methods to try to give a more of an idea, we come into the sample usually with a bigger X-ray beam size. And when we have these near field detectors, the detector slots in very closely behind the sample. And the position and shape of the diffraction spot on the detector, it depends on where is the grain inside the sample. When we have these far field detectors, the detector's very far back, and the position of the spot on the detector, it depends just on the scattering angles. So just on the kind of internal chemical crystallography and unit cell. And by putting these two things together, the idea is to get a complete picture of what happens inside the sample. So a first example I'll take is the PhD project of Michaela Labella. So she completed her PhD, I think, last year, and she's a postdoc now. So she successfully managed to do research through the COVID pandemic. And in this case, there was the combined work that first there's the X-ray tomography, or let's say at the same time, there's the X-ray phase contrast tomography. And you could watch this chemical reaction. So this is basanite hydrating to gypsum. You could watch the shapes of the crystals changing, and you could watch that in 3D and in detail. And the questions they had was, how did this reaction go? And were there orientation relationships and structural relationships between these two chemical compounds? So they could watch in tomography how the crystals transform. And then this scanning 3D XRD method was then to understand which crystal had which crystal structure and how were they transforming, and to look for the different phases inside and so on. So another example with the grain mapping methods, this time from metallurgy, from Eunice El-Hatchi, Sophie Bervier and co-workers. So this is a shape memory alloy. It's a copper aluminium beryllium in this case. And so when you stretch or deform this alloy, it has a large elastic response, and then eventually it goes through a phase transformation, and that gives this famous shape memory effect, where you can deform the metal and then heat it up and it will come back to its original shape. So understanding how that works in detail, they've done this 3D XRD experiment where they measure all the diffraction angles. And from the diffraction angles, they have the grain orientations. They have something about the chemistry in the grains, and they have the stress and the strain tensors in the grains. And the sample is inside of a load frame. So as they apply load to the sample, they can see each single crystal, how it responds elastically. The diffraction contrast experiment, they can see the shapes of the grains, and they put all of this together with a finite element calculation to understand what really happens in the microstructure. So we see this idea of taking the diffraction data together with the detailed simulations to put everything together to try to understand what happens inside the material. So I come on now to one of the most shall we say, high resolution methods we have for mapping in 3D looking at grains. This is called topotomography. This is something where Wolfgang Ludwig has been a key developer, and recently, together with Henri Proudhon, they've adapted all of the macros that drive the diffractometer to do this. So they work with our new control system, and they're all optimized for bliss. So at the top, you see how the diffractometer is moving, and it's rotating around a scattering vector. And that's the scattering vector of this particular crystallite, this particular grain that's inside the sample. Now the diffractometer's gone to find another reflection or another grain. And so the trick here, which is easier said than done, is that you rotate around the scattering vector of one of these crystals and you get this beautiful picture where you see all the slip bands inside. And then you go and find the neighboring crystal that's right next to it, and you do the scan again for the neighboring crystal. And if you succeed to do that, and in this case they did, then you succeed to see the slip bands transferring from one grain into the other. So you can understand how slip transfer works across grain boundaries. So I would say this is a tour de force in what you can do with carefully driving the diffractometer. And we look forward to seeing these kinds of things, of studying specific grain boundaries in detail, becoming more common in the future. So for all of these things I've explained with diffraction and imaging combined, we have this notion of there's an inverse problem. And I'm very happy that it's Tillman that's introducing me here, because Tillman obviously has been working on these with the texture tomography aspect. In the ID11 case, we usually think about this somehow as there's a 6-dimensional problem. So you have a 3D sample, and we imagine for each voxel in the sample, we have a 3D diffraction pattern. And from this 3D diffraction pattern, you may then want to pull out all kinds of different information. And so we have this suite of different methods on the beamline that you can use depending on what kind of information you want to pull out and depending on how you think you're going to be able to get the information out of the sample. So the one I just showed, topotomography, you have a full 3D image of the sample, but you only look at one Bragg diffraction peak. So that's very good for imaging things in space on one particular grain. The DCT grain maps that I'll show later, you have like a 3D data set, and you have this 6-dimensional problem of you need like the orientation for each of these voxels. So because it's only 3D data, but with a 6D problem, this works really well when the orientations are quite discrete. So when the sample is undeformed and each crystal in the sample has just one unique orientation, then you can overcome this kind of missing data problem. As the sample becomes more and more deformed, this is especially for metallurgy, but this works in the same way for all the different kinds of samples we see. As the sample becomes more deformed, then we try to make the beam smaller. So we can take slices. There's this kind of HEDM or 3D XRD method where you do 2D slices and try to reconstruct the grain maps. And what's growing a lot recently is this XRD-CT or scanning 3D XRD approach. So in this case, we scan a small beam across the sample and we do a rotation at each point. So we have this kind of 2D scan of position and rotation on the sample and a 2D diffraction image at each point. So we have this kind of 4D data set and we're reconstructing a 5D problem, which is this like diffraction pattern at each point. So at this point, we're only missing one dimension. So we're, should we say, almost respectable. We can get back in the case where the sample has some symmetry and be fully respectable. So in this case of glassy samples, this works fantastically well. So this is a bulk metallic glass that Mihai Stoica and co-workers brought to the beamline. And in this case, it's a metallic glass that segregates like oil and water, so something like a salad dressing. And you could resolve the different components inside the glass by looking at the X-ray diffraction data. And you could scan across and see inside this material, using the X-rays, how you had the phase segregation and how you have obviously different length scales. And you can see which side of the sample was quenched and which side wasn't quenched. And there's, as always, much more details we could talk about, like what's happening around the little crystals inside of the sample. The same idea of doing XRD-CT on glass is this time was for strain extraction. This was with Sergio Scudino and the shear bands that he finds in glasses. So in this case, we fit the position of the diffraction ring around the circle, and the strain tensor component that's parallel to the rotation axis can be reconstructed. And this gives you a contrast that you can see in a piece of glass where it's undergone this mechanical strain. And there's, I think, a very interesting research question of what happens when you reconstruct the component which is in the plane, which you shouldn't actually reconstruct because you're rotating the sample and you measure different things in different directions. But nevertheless, it gives a very nice picture. So I'll try to get towards the wrap up with the strain tensor reconstruction we're doing most of, which is by these 3D XRD methods. So in this case, it's the first example that I'm aware of doing on the nanofocus station at ID11, and it was a tin whisker that Johann Hector had fibbed out. So he found a tin whisker starting to grow. So it's a piece of copper with a thin layer of tin, with tin on the surface. And these whiskers grow and cause all kinds of problems, and they wanted to understand what was happening there. So he selected out an area where a whisker was growing, and we have this 3D scanning grain map, this 25 microns across the sample, so it's submicron resolution, and there's the hydrostatic stress. So they tried to get out the stress and the strain as a function of position across these grains. And you get, of course, the detailed shapes of all the grain boundaries, you get the misorientations of all the grain boundaries, you have a wonderful richness of data. You see all of the secondary phases and so on. And so we see this as a method that can be applied to many different problems in the future. For the reconstruction of that data, there's a lot more progress that's been made in recent years, especially by Axel Henningsen, who's just finished up his PhD in Lund, on the different ways that you can reconstruct these, what I call type 2, type 3 strain fields. So type 2 is the variation from one grain to another grain. Type 3 is the variation inside of a grain. And then there's actually a type 1, which is the thing that we've been doing for many years, which is just a general strain map across the whole sample. So where things have changed, let's say in the last 5 years, is where we used to just do a strain mapping across a component. We now start to do a strain mapping from grain to grain and a strain mapping inside of the individual grains to cover all these different length scales. So I won't say very much about how long it takes to collect data. Suffice to say that it depends a lot on your sample and how you do the scans. But reconstruction of the data is usually a much bigger problem than collecting it. And so there's a big progress for us that Haijing has written up nice documentation to make those reconstructions go faster. I'll finish up with showing a video of our kind of fastest grain mapping method for 3D at the moment. So this is the case of undeformed grains. They're recrystallized grains that are growing. This is actually an experiment from before EBS, and it's the PhD of Jin Zhang. And they had 15 time steps, and they follow the evolution of the microstructure in this pure iron. So you see there's these very, very complicated grain shapes, and you have a movie of how all of these grain shapes are changing as a function of time. And there's then some very detailed analysis because you have more than, I think, 13,000 grains. You have many, many, many different grain boundaries. And the question is which grain boundaries are moving in which directions and why? And something I look forward to seeing in the future is to do these kinds of experiments continuously as in situ experiments without having to interrupt. In this case, when the data were collected, we had to stop the experiment, interrupt, and then measure. And I think it would be wonderful to do this experiment as a continuous movie in 4D in the future. So with that, I'd like to thank my team that helped me and that do all the actual work at ID11. Here are some of us enjoying our Christmas dinner. And I thank you for your attention. And I look up and I don't see the audience. So I hope you're all looking up and still following. So thank you.
Well, thank you, John. Thank you for that really nice talk. I'm opening the floor for questions. As we're quite a lot, I would ask you to type your questions in the Q&A box below so I can read them out and we get an answer there. So yeah, I saw some hands coming up. While people are talking, I would like to go back to you and ask a little bit about EBS and what's the good that it brings for you. So which kind of experiment do you think are feasible now that were not feasible in the past? What is your kind of dream experiment? And then also the downsides of EBS. How are you dealing with all this data that the IGA is spitting at you?
So the dream experiment for me started in 2016 with this nanofocus station. And so I think I remember when you were around, we had Marta and Pavel were the postdocs. And we spent hours trying to get this diffraction tomography experiment to work. And I think the example I showed from Johan of the tin whisker was the main paper to come out from that time. And we had a really hard time with drift, that we weren't quick enough to measure before the sample moved. And now when we do the experiment, I mean, in terms of flux, we're say between 10 and 40 times higher flux. The detector is 100 times faster with the Eiger. So now we're always driving our rotation axis at full speed. So this scanning XRD-CT, this is really working now, and it was something we were really struggling with before the EBS. So that's been a complete game changer for us, that experiment now routinely works. As you rightly point out, the data processing is very heavy. I think the last time I did a webinar was with the HDF5 European users meeting. So this business of running the data through a GPU and finding fast ways to process these large volumes of data, it's a lot of fun for me, but I think for user groups, it's probably still frustrating or overwhelming that you can't take the data home with you. So we tend to run the data reduction on the cluster here, try to get as much done in 3 months as possible, and try to get to the point where you have the publication quality figures and the results as soon as possible. But this is now, I think, data processing is the bottleneck.
Yeah, I agree with you from my own painful personal experience. There's still no questions popping up, so I just go ahead and ask the next question. What is it in terms of user proposals that you're welcoming now? Do you see like trends in which kind of experiment people are putting to the beamline? I mean, of course, you cannot talk about the proposal in detail, but which flavor of experiment is coming up now, and what is where you see the beamline going?
So the general trend that I see is that across all the disciplines, so from the crystallography to the material science kind of microstructures, across all of our disciplines, the X-ray experiment now needs to be matched up with some modeling. So when I started doing research, you could do a crystal structure, publish the crystal structure, and that was kind of a complete story in itself. I think now if you publish an interesting crystal structure with interesting chemistry in it, you'll need a DFT calculation to go with it. If you look at the grain growth experiments, then together with it, there's a phase field model, and there's a detailed study of what you expected all those grain boundaries to be doing. And I think the challenge for us in the coming years is that we need to bring the X-ray experiment and the X-ray data to be much closer to the questions in modeling. I can expand on that, but if there are questions in the chat, then...
I mean, I see raised hands in the chat, but I don't see the questions. So Aditya, perhaps you can write the questions in the Q&A. The same for Benoit. Or perhaps I'm not good at.
Yes, please, because in this webinar we cannot open the microphone to the audience, so please just write down Benoit Mallard and Aditya Shukla. Please write your question in the question and answer box, please.
So we have the first question from Manuel Marechal. He thanks you for presenting these very interesting results, and he wants to know what's the lowest time and space resolution up to now. And his question concerns potential uses in the soft matter materials, the size of dynamical grains ranging from a few 10 nanometers to a few 100 nanometers.
So thank you for the question. So the lowest time resolution for us at ID11 normally, I would say, is the speed of the Eiger detector. So this usually goes up to 500 hertz. And I think you can get it in a mode that goes to 1 kilohertz. But in that case, you just get one single diffraction pattern, so you don't get the full image. You can go to much faster time resolutions on time-resolved beamlines, of course. For soft matter, I think Tillman is probably the better person to ask. there's a difference in X-ray diffraction, that things kind of fundamentally change when you go from tens of nanometers up to a few hundred nanometers. So the amount of peak broadening that you get from the crystallite size suddenly kind of jumps, or it goes as kind of 1/4 crystallite size somehow. So if you have a few hundred nanometer grain size and it's something nice like platinum or gold, then we get very nice diffraction spots, and we can use these spotty diffraction methods. If you have 10 nanometer size grains, then we have these things that look like smooth powder rings. And so it completely changes the way you would do the experiment. So for ID11, we can reach about 150 nanometers, I think, and that comes from the run out of the diffractometer as well as the beam size. I think you can go to smaller beam sizes in other beam lines, especially for soft matter. I'd look at ID13, I think.
Yeah. Yeah. I mean, I'm just adding that ID13 has smaller beams for that, but we have a separate webinar coming up on ID13 in the near future. There's another question by Aditya Shukla. Is there a limit on how big or small the single crystal can be in the high pressure experiment?
So in the diamond anvil cells, if the crystal is bigger, they just won't bring it to the synchrotron because you can do it on a lab instrument. The limit for how small it is if somebody can find that there's a single crystal in the sample and that they're not looking at a powder diffraction experiment. So I would say there's this just right size, which is in the range of around a micron or a few hundred nanometers, where you actually have a single crystal, but it's too small to do in another beamline. But if the crystal's large, then you just wouldn't bring it to us because it's too difficult to get time with us, I think. I'm not sure that answered.
The next question from Benoit Mala is if it's possible to obtain chemical information like volume fraction of oxygen or nitrogen, for example, in 3D at the grain size.
So hi, Benoit. So something I glossed over in the talk, what we actually map when we do these experiments is the unit cell parameters across the sample. So if you can get the composition by looking at the unit cell parameters, like what we would call D0, then you can pull out that kind of information. Something that we're working on at the moment that is not, let's say, ready for general consumption would be to look at the diffracted intensities and do a structure refinement as a function of position in the sample. Personally, I think that would be fantastic when we can get that to work. As of today, I'm not ready to show any results. And I think if you look for, say, oxygen or nitrogen in steel, it would be a difficult one. But in general, to refine the crystal structure as a function of position in the crystal, I think is one of the directions I'd like to go in the future. And I would welcome any proposals to come and try and get that to work.
Great, thank you. So there is no further open questions coming up here. I guess I would like to take the opportunity to announce the next webinar, which is going to be ID29, a protein crystallography talk on the 15th of February, so just a week after the user meeting. And until then, I hope I see a lot of you at the user meetings in 2 weeks. I look forward to the discussions, and I once again thank John for this very interesting talk, and you, of course, for attending it. Thank you.
Thank you very much. Thank you again, John, and thank you, Tillman. So now we can stop recording maybe, and